In this video, I'm going to be talking about the signaling that occurs through the beta-adrenergic receptor as part of the fight or flight response. You're probably familiar with this. This is the physiological response to acute stress or a dangerous situation. For example, rounding a bend in the trail and coming face to face with this big guy. The fight or flight response prepares the body for battle or to run or escape. It's mediated by the hormone adrenaline, which causes an increase in heart rate, pupils to dilate, and a whole host of other responses that are designed to help you survive. One of these responses is to mobilize stored glucose and fat for cellular energy. In other words, for ATP production, so that you can outrun that bear. So how does the hormone adrenaline cause tissues like liver and muscle to increase energy production? To answer that, we have to zoom in to the cellular level. The hormone adrenaline is produced by the adrenal glands and released into the bloodstream to interact with target tissues throughout the body to achieve the response. Adrenaline is also known as epinephrine, and those two terms are used interchangeably. Circulating adrenaline encounters its receptor embedded within the cell membrane on target tissues. For this response, the tissues that we're interested in are liver and muscle cells. So here's the adrenergic receptor embedded within the phospholipid bilayer. This gray rectangle represents the cellular membrane. And when adrenaline binds to the adrenergic receptor, the receptor becomes activated. Now, this is a G protein coupled receptor, and as we've discussed before, the typical mechanism by which G protein coupled receptors achieve their response within the cell is by interacting with intracellular G proteins. And here's that heterotrimeric G protein that interacts with our adrenergic receptor right here. Once the receptor becomes activated, that causes the G protein to do something called GDP-GTP exchange. So this GDP that the inactive G protein was holding is released, and a brand new shiny GTP from the cell is picked up and bound instead. And that's going to activate this G protein subunit to dissociate from the other two subunits, slide along the membrane, and interact with the downstream target enzyme. Now, in this pathway, that downstream target enzyme is an enzyme called adenylyl cyclase. Adenylyl cyclase, in turn, once it's activated by binding of this G protein, synthesizes cyclic AMP, which becomes the second messenger for this signaling pathway. As we've talked about before, second messengers are small diffusible molecules that are not proteins, but what they do is they transduce the signal throughout the cell, right? So a signal that was up here at the membrane is now being transduced throughout the cell. And that's actually what's going to lead to the cellular response, is the interaction of this cyclic AMP second messenger with downstream target enzymes. But before we talk about that, let's take a closer look at this cyclic AMP molecule and see how its synthesis and degradation is controlled in the cell. So here in the center, we have our cyclic adenosine monophosphate, or cyclic AMP, and it is synthesized by that activated adenylyl cyclase enzyme from ATP. So we've seen this ATP molecule before. Here's our adenosine portion of ATP, the sugar and the adenine nitrogenous base, and here are our three phosphates, so adenosine triphosphate, ATP. And what this enzyme does is it cleaves off the two terminal phosphates from ATP and then cyclizes the remaining phosphate to the sugar ring. And we can see that right here. That one remaining phosphate group is now connected to the sugar ring in two places. And that's where we get that name, cyclic AMP. Now, cyclic AMP is in turn broken down by an enzyme called phosphodiesterase. And what phosphodiesterase does is it cleaves that second bond to the sugar, and what we end up with is plain old vanilla adenosine monophosphate, AMP, which is not a second messenger and does not activate those downstream cellular responses. And so Phosphodiesterase then acts as an off switch to remove cyclic AMP from the cell and turn off this signaling pathway. So the concentration of cyclic AMP is controlled by the competing activities of these two enzymes, adenylyl cyclase, which is increasing the concentration of cyclic AMP in the cell, and phosphodiesterase, which is degrading and decreasing the concentration of cyclic AMP within the cell. And so phosphodiesterase is always degrading this. The only way we're going to have an increase in cyclic AMP concentrations is if we're activating adenylyl cyclase downstream of that adrenergic receptor. Now, cyclic AMP is a very commonly used second messenger, and it's capable of influencing many cellular signaling pathways. But in the context of this specific response, it influences the activity of an enzyme called protein kinase A, or pKa. Here's the structure of pKa. It has 4 subunits, 2 of which, indicated in red here, are actually the enzyme domains, and actually the kinase domains of the protein. And then down here in blue are the regulatory domains. And more specifically, these are inhibitory. So when the inhibitory domains are bound to the kinase domain, this is an inactive enzyme. It has no kinase activity. So what happens is that when cyclic AMP levels rise within the cell, they bind to those inhibitory subunits. And when cyclic AMP is bound, there's a shape change in those inhibitory subunits, causing them to release the kinase domains, which now become activated. And as we know, kinases phosphorylate target proteins, and so that's exactly what's going to happen. These activated protein kinase A kinase domains are now going to phosphorylate their targets. So in the context of this response of mobilizing energy within the cell, what are those target proteins? The first target of this activated protein kinase A is an enzyme called phosphorylase kinase. So kinase A is going to add a phosphate group onto this enzyme, now causing this enzyme to become activated. And as we can tell from the name, the activity of this enzyme is also going to be a kinase activity, a phosphorylation activity. The target of phosphorylase kinase is an enzyme called glycogen phosphorylase. Get it? Phosphorylase kinase phosphorylates glycogen phosphorylase. So it's going to add a phosphate group to this glycogen phosphorylase, thereby activating this final enzyme in our cellular signaling pathway. And this is actually the enzyme that's going to yield the cellular response in liver and muscle cells and cause the release of energy. So what does this enzyme do? Well, the name kind of gives us a clue. Glycogen phosphorylase interacts with the glycogen granules. There's that beautiful structure of the glycogen granule we saw before, all these long chains, many branched chains of glucose molecules, tens of thousands of these glucoses. And this enzyme is going to start attacking these glycogen chain ends and releasing glucose into the cell, causing the concentration of glucose to start to rise dramatically. And what happens with glucose when it's released into the cell? Well, now it can be used in glycolysis and cellular respiration to generate cellular ATPs, which can fuel that muscle contraction that's going to be required so that you can outrun that bear. Phew! And now remember that concept that we talked about before of signal amplification. At each step of that pathway, multiple enzymes are going to be activated. So we're going to have lots of copies of this glycogen phosphorylase attacking at all these ends and really mobilizing a ton of glucose for the cell to use. So once you've survived, how is this pathway terminated? Well, it's terminated at multiple levels. First of all, once the danger has passed, the adrenals are no longer going to be secreting adrenaline into the bloodstream. And so the concentration of adrenaline is going to go down, and that means that the adrenergic receptor on those liver and muscle cells are no longer going to be bound by adrenaline. So the receptor is actually going to be inactivated. The second level at which this is terminated is at the level of the G protein, which, as we've talked about before, has that GTPase activity and will spontaneously hydrolyze the GTP that it's holding. And when that GTP is hydrolyzed, now that G protein returns to its inactive state, so it'll no longer be activating adenylyl cyclase. Now, if adenylyl cyclase is no longer activated, it's not going to be producing cyclic AMP, and we also know that that enzyme, phosphodiesterase, is going to be cleaving any existing cyclic AMP in the cell to yield plain old AMP, adenosine monophosphate. And then finally, we know that for every kinase that exists in the cell, there are going to be corresponding phosphatases that remove those phosphates that were added to those enzymes, activating them, and that's going to lead to inactivation of those kinases, thereby terminating our cellular response. All the protein structures that I used in this video were obtained from the Protein Data Bank, and you have the references here. And the glycogen structure is one I've used before by Michael Hagstrom.